Hi everyone, my name is Hsp70 and I will be presenting the work that I have done regarding Hsp70, which is a molecular chaperone and its interactions with a full-length substrate with members of the Garage Lab at the University of Massachusetts Amherst. The Hsp70 chaperone system is a vital component of the quality control machinery of a cell. It maintains the integrity of the proteome and it's conserved across all domains of life, from bacteria to human. DnaK, the E. coli ortholog, which is most abundantly studied out of all the Hsp70s, is comprised of a substrate binding domain, the SBD, and a nucleotide binding domain, the NBD, which is connected by a flexible linker. The SBD is known to bind short hydrophobic sequences flanked by positively charged residues. Below is a figure of the beta SBD in green and the alpha helical lid in blue, which opens and closes to accommodate the substrate. Bound in pink is the NR peptide. Historically, most studies have used peptides like NR to understand DnaK's mechanism of action and substrate binding, but it doesn't quite paint the full picture, as most Hsp70 clients are mostly nascent polypeptide chains or ones that are unfolded under cellular stress. Therefore, my goal is to understand how DnaK interacts with full-length substrates, and I aim to answer questions like what sites are bound by DnaK when it's presented with multiple potential binding sites, and does DnaK bind all these sites presented in a full-length substrate with the same affinity? And are there regions on the full-length substrate that DnaK binds aside from the already predicted ones? I hypothesize that DnaK, through its canonical binding site, interacts with short hydrophobic sequences within the full-length substrate. In order to study DnaK's interaction with a full-length substrate, we chose alkaline phosphatase A, which is shown to be an endogenous substrate of DnaK. It's synthesized as a precursor and is then translocated to the periplasm of the E. coli, where it matures to its 471 amino acid self as a dimer. It is shown to be experimentally well behaved, as it is stable and soluble under reducing conditions that eliminate 4 native disulfides. And 5 DnaK sites were also identified on proPhoA in a peptide array and are shown in the figure on the left and are labeled as A, B, C, D, and E. We are using large fragments of phoA, which I will refer to here on out as phoA1, phoA2, and phoA3, as a tool to closely dissect the binding of DnaK to this substrate and to simplify its characterization with DnaK through NMR spectroscopy. Each fragment is approximately 100 amino acids long and contains 1 to 2 binding sites. phoA1, the first fragment, contains binding site A and B, A being in signal sequence of the precursor form of this protein. phoA2, the second fragment, contains binding site C, and phoA3 contains binding sites D and E. The construct used to purify these fragments was generously gifted to us by the CalDemos lab. It contains an N-terminal His tag followed by MBP that has a respective fragment fused to its C-terminal tail. MBP stands for maltose binding protein, and it also increases the stability and solubility of the fragment. There is also a cleavage site for tobacco etch virus, TEV protease, for the separation of the fragment from MBP. This construct was overexpressed in BL21 cells and purified using nickel immobilized affinity chromatography. This step separates the MBP fragment fusion from the rest of the cell lysate. A TEV cleavage follows this step, and then we take advantage of the His tag again to separate MBP, TEV protease, which is also His tagged, and any remaining uncleaved fusion protein to obtain pure fragment. I usually follow this with gel filtration to remove any remaining impurities. The sample was then exchanged into a buffer suitable for NMR, and the sample contains 40 micromolar ILV labeled DnaK SBD and unlabeled proPhoA fragments that are at 30 micromolar for fragment 2 and 50 micromolar for fragment 3. And the spectrum is obtained at 25 degrees Celsius with 5 millimolar ADP. This figure displays each of the series of steps in the purification described previously. The first lane contains the cell lysate, which is separated from the MBP fragment fusion, which elutes with 400 millimolar imidazole. The fusion protein weighs about 55 kilodaltons and also has bands present immediately beneath it, which are indications of endoproteolytic cleavage. The third lane also shows products resulting from cleavage. As you can see, TEV cleavage is not 100% efficient as there still are remnants of the fusion protein at 55 kilodaltons. But at the bottom, cleaved fragment is also visible around the 13 kilodalton mark. After a second round of nickel affinity chromatography, we expect that MBP, TEV, and the fusion protein to elute with imidazole and the pure fragment to elute in the flow-through, as can be seen in the last lane. The overall yield of the pure fragment is very low. During the purification of phoA1, an interesting result was observed. During the second nickel affinity chromatography, which is expected to separate MBP, TEV protease, and the fusion protein from the fragment, I observed the presence of an extra band in the flow-through along with phoA1, which is boxed in yellow at the bottom of the gel. This extra band is endogenous DnaK, which weighs around 70 kilodaltons. This result strongly suggests that DnaK sustains an interaction with the fragment phoA1 during purification, demonstrating that it has a high affinity for this first fragment, which contains sites A and B. There is a band also immediately underneath the 70 kilodalton DnaK band, which is suspected to be slight endoproteolytic cleavage of the chaperone. On the right 4 bands, you do see MBP and TEV protease eluting in high imidazole concentrations of around 400 micromolar, which is expected. In order to characterize the interaction with DnaK and the fragments, the methyls of isoleucine residues in the SBD were labeled, and coupled with transverse relaxation optimized spectroscopy, or the TROSY technique, to obtain high-resolution spectra for proteins with higher molecular weight, such as the SBD in complex with fragment. Labeling the isoleucines gives insight into key residues in the binding cleft of the SBD, which contains 2 diagnostic isoleucine residues. Their chemical shift perturbations, or CSPs, directly report on substrate binding. As you can see on the figure on the right, in black is a spectra for the empty SBD, which is overlaid with the SBD interacting with peptides C, D, and E. C is in green, D is in blue, and E is in purple. Each peptide has a different methyl resonance fingerprint, and there is slight peak splitting visible for peptide E in both isoleucines. This is a spectra of the empty SBD overlaid with SBD in complex with phoA2 and SBD in complex with peptide C. The red peaks indicate that SBD can be found both in the empty state because it overlays with the black peaks, and when it's in complex with a fragment, it binds using the same site as is presented on the individual peptides, and that can be seen because the red peaks overlap with the green peaks representing SBD in complex with peptide C. Similarly, this is a NMR spectra of empty SBD in black overlaid with an SBD interacting with fragment phoA3 overlaid with its constituent peptides D and E in blue and purple respectively. This spectra indicates that DnaK definitely binds phoA3 through multiple sites other than the predicted D and E. This spectra overlays empty SBD with SBD in complex with phoA2, phoA3, and full-length proPhoA respectively. It suggests that DnaK binds full-length protein using the sites also found in both the fragments, but it also binds sites presented outside of these 2 fragments as indicated by the cyan stars in the downfield region of the spectra. There is also a peak present in fragment phoA3 upfield in the proton dimension that is not overlapping with any other peak. It is tempting to speculate that this is a new site that DnaK is binding when only presented in the fragment phoA3. Overall, we can conclude that DnaK has a high affinity for fragment phoA1 based on behavior during purification, and the NMR spectra can help us conclude that the binding of SBD to phoA2 occurs via the same mechanism by which the SBD binds the individual peptide C, and the interaction between fragment phoA3 and the canonical binding site of SBD is not completely identical to the binding between SBD and the short peptides. Moving forward, I would like to explore the interaction between chaperone and fragment phoA1 through NMR, as that was not yet achieved, and test weaker binding sites by ordering corresponding peptides. I can also ascertain the affinities for each peptide sequence and its corresponding fragments via a competitive binding assay. Furthermore, I would like to thank the members of the Garrosh Lab for their valuable discussion and the Kalajimos Lab for kindly providing the proPhoA fragment constructs. Thank you so much for listening.